Historically, we developed a technique more than 15 years ago, at least the first generation of a technique, that would allow us to look inside the intestine of a newborn baby, human baby. And unbeknownst to many people, newborn babies have all kinds of complications, either because of premature birth or shortly after birth. They develop digestive problems. There's sometimes chronic inflammation of the intestine. And the reason why the intestine is also very important is at the time of birth, it is a point where the human body becomes populated with microbes. And this is because the baby starts to actually consume breast milk immediately following birth. And their food drives the populations of microbes that begin to flourish in the baby. And that's part of normal developmental biology. That's the way we've been operating as humans for thousands of years. So the question is, how do you characterize the impact of nutrition on a newborn baby? Newborn babies, especially if they're healthy, you can't cut them open, you can't biopsy them, it's hard to even get blood. So you have to develop a non-invasive technique if you're going to understand the human biology. And that really was part of our goal, to develop a non-invasive technique for assessing the biology of the human intestine. So in this paper we decided to look at premature babies that are born in a certain range, so they're highly premature. They're in neonatal intensive care units. I have a collaborator there, Dr. Cami Martin, and so she is a neonatologist, again, a faculty member at Harvard, and she provided us with fecal samples, stool samples from babies in that intensive care unit. They have very complex biologies because they're very premature. But they don't know what kind of nutrition to give those babies and whether or not they're doing inadvertently some harm to the baby. So that's important. Then another collaborator, Dr. Sharon Donovan at the University of Illinois in Champaign, she's a pediatric nutrition expert, so she understands the nutritional needs of babies, and she got us full-term babies access that were either exclusively breastfed for the first 3 months of life or exclusively formula fed for the first 3 months of life. Then we did our diagnostic, and our diagnostic is unique because, again, it's non-invasive, and it isolates material, genetic material, from the cells that are actually passed out in the fecal stream. We capture the RNA, ribonucleic acid, from those cells and do something called deep sequencing. We actually sequence the signature of the RNAs in that material. And we said, what genes are there? What are these babies, their intestines, actually producing in terms of gene expression? And the whole idea is we can take these genes and basically use them like a map, a constellation, in order to determine how the baby's biology is changing over time. So we compared the two, the highly premature versus the full term, and we found some very striking differences, which isn't terribly surprising. But this study is a proof of principle that we can non-invasively monitor the biology of the intestine and the immune system in these highly premature infants and full-term infants and begin to unravel the developmental processes that are, in one case, going along just fine, that's the healthy babies, and the other case, the premature babies, not going along very well. Then, of course, the idea is to use this information in order to optimize treatments to help these babies. After all, every mother and father want the healthiest baby possible. They want a baby that thrives. They all want brilliant babies, So a lot of these developmental processes actually impact neural development and other components. And we know enough about biology now to understand that if there is if there are serious disruptions in this early developmental process, they can have impact for the entire life of that human.